Hello and welcome to BiG Science Academy. I am Bikram Ghosh. So let's continue with respiration. Today our topic of discussion is Krebs cycle. So let's begin the discussion. So at first, let's see the key points of this video on which we will discuss in this video. The first point or the first topic will be the link reaction, and the second point or the second topic will be the Grape cycle or citric acid cycle. So let's start. So let's begin with the link reaction. In the previous video, we have seen that in the glycolysis process, the respiration process starts. That means the total respiration process starts with the glycolysis step. And in this glycolysis step, the respiratory substrate, which is the glucose, is converted into two molecules of pyruvate. And now, these two molecules of pyruvate are converted into two molecules of acetyl-CoA in the link reaction process or the link reaction step. And this process or reaction occurs with the help of an enzyme which is known as pyruvate dehydrogenase complex. And in this reaction, a NAD plus molecule is converted into NADH molecule and a CO2 molecule is released. That means these two pyruvate or the two molecules of pyruvate, which is a 3-carbon compound, is converted into a 2-carbon compound which is acetyl-CoA. So let's see some important points of this link reaction. At first, we can see here that it is an irreversible process. That means the conversion of pyruvate to acetyl-CoA can take place only in a unidirectional way. That means once the pyruvate is converted into acetyl-CoA, it cannot be go backward to form pyruvate again. So it is an irreversible process. Next, it occurs or the link reaction occurs in the mitochondrial matrix of eukaryotes and in the cytoplasm of prokaryotes. So in eukaryotes, we know that there is a mitochondria organelle, and inside that mitochondrial organelle, the matrix is present and the link reaction occurs in that matrix only. And in the cytoplasm or in prokaryotes, the cytoplasm is the site of link reaction, since there is no organelle present in prokaryotes. Next, the third point is that it is an oxidative decarboxylation process. That means in the link reaction, decarboxylation occurs. That means one carbon is removed from a reactant to produce product, or carbon is removed from a substrate to produce the product. Here, the pyruvate is a 3-carbon compound which is converted into a 2-carbon compound acetyl-CoA. That means there occurs an elimination of carbon in the form of CO2. That's why it is known as oxidative decarboxylation process. And the last or the most important point of this link reaction is that the enzyme which is helping for this link reaction is the pyruvate dehydrogenase complex enzyme, which is a multi-subunit enzyme complex. That means more than one enzyme acts simultaneously to produce these acetyl-CoA molecules. So the first complex or the first unit of this multi-subunit enzyme complex is the E1 or the enzyme 1, which is pyruvate dehydrogenase. And the second one or E2 is the dihydroxylipoyl transacetylase. And the last one or the E3 enzyme is the dihydroxylipoyl dehydrogenase. These three enzymes act together on the pyruvate molecules to convert it into acetyl-CoA and to convert the NAD plus molecule to NADH and to expel or eliminate the carbon in the form of CO2. So this is the total link reaction where this pyruvate molecule is converted into acetyl-CoA with the help of pyruvate dehydrogenase complex. So now we will discuss about Krebs cycle. So from the start of respiration, we know that glycolysis occurs, and from glycolysis, two molecules of pyruvates are produced. And then these two molecules of pyruvates are converted into acetyl-CoA by the process of link reaction we have just seen. And then these two acetyl-CoA molecules enters into the Krebs cycle. Okay, so now we will discuss about the Krebs cycle. What is going in that process? So let's see the important points of this Krebs cycle. First, the Krebs cycle is also known as tricarboxylic acid cycle or citric acid cycle. Second, the Krebs cycle is an oxidative decarboxylation process, just like the link reaction. That means here also elimination of carbon occurs in the form of CO2. That's why it is known as decarboxylation process. The third point is that it occurs in the mitochondrial matrix of eukaryotes and cytoplasm of prokaryotes. Just like the link reaction also, this Krebs cycle process occurs in the mitochondrial matrix of the eukaryotes and cytoplasm of the prokaryotes. And the fourth point is that substrate level phosphorylation occurs in the Krebs cycle. That means direct ATP production occurs in this Krebs cycle process. And the last point is that the total number of ATP molecules produced in a single cycle of Krebs cycle is 10, which is produced for one molecule of acetyl-CoA. So in total, 20 molecules of ATP are produced for the 2 molecules of acetyl-CoA produced by the link reaction process. We have seen that in the link reaction process, 2 molecules of pyruvate are converted into 2 molecules of acetyl-CoA. So for each acetyl-CoA molecule, 10 ATP are produced, and for 2 acetyl-CoA molecules, total 20 ATP molecules are produced at the end of the total Krebs cycle process, which takes place 2 times. So now let's discuss about the Krebs cycle in an elaborative manner. At first, the pyruvate molecules, which is a 3-carbon compound produced at the end of the glycolysis, is converted into acetyl-CoA in the link reaction process. It occurs with the help of the enzyme pyruvate dehydrogenase. And in this process, we have just seen that a NAD plus molecule is converted into NADH molecule. And decarboxylation occurs, that means elimination of carbon occurs in the form of CO2. And a CoA molecule or coenzyme A is added to this reaction for the production of acetyl-CoA. And the end product of this link reaction, which is acetyl-CoA, is a 2-carbon compound. So from now onwards, the actual Krebs cycle will start. So the link reaction product, which is acetyl-CoA, at first enters into the Krebs cycle and combines with the oxaloacetic acid, which is a 4-carbon compound, to form this citric acid with the help of an enzyme which is citrate synthase. And here the CoA of the acetyl-CoA is eliminated out. And the product which is formed, which is citric acid, is a 6-carbon compound. And this citric acid is the fast stable intermediate of the Krebs cycle. And that's why the Krebs cycle is also known as citric acid cycle. So now the 6-carbon compound, which is citric acid, is converted into isocitric acid, which is also a 6-carbon compound, with the help of an enzyme aconitase. Now this isocitric acid, a 6-carbon compound, is then converted into a 5-carbon compound which is alpha-ketoglutaric acid with the help of the enzyme aconitase again. And in this step, a NAD plus molecule is converted into NADH and decarboxylation occurs with the release of CO2 molecule. That means this decarboxylation step is needed for the conversion of a 6-carbon compound to a 5-carbon compound. That means from the 6-carbon compound, 1 carbon is being eliminated in the form of CO2 to convert the 6-carbon compound into a 5-carbon alpha-ketoglutaric acid molecule. So now the alpha-ketoglutaric acid is then converted into succinyl-CoA with the help of an enzyme alpha-ketoglutarate dehydrogenase. And here also a NAD plus molecule is converted into NADH molecule. And here also the elimination of CO2 occurs. That means here also the elimination of carbon occurs to convert a 5-carbon molecule into a 4-carbon compound. And to produce the succinyl-CoA molecule, a coenzyme A is added to this reaction. Next, we will see that the succinyl-CoA molecule, which is a 4-carbon compound, is then converted into succinic acid, which is also a 4-carbon compound. But here, the coenzyme A is released and which is again utilized back to produce succinyl-CoA. And this conversion of succinyl-CoA to succinic acid occurs with the help of an enzyme succinyl-CoA synthetase. Here it is synthetase, and in this step here it was synthase. So keep that in mind because students actually gets confused with synthase and synthetase. So in this step, substrate level phosphorylation occurs. That means direct ATP production takes place. Here ADP molecule is converted into an ATP molecule. So now we will see that the succinic acid molecule, which is a 4-carbon compound, is then again converted into a fumaric acid molecule, which is also a 4-carbon compound with the help of an enzyme succinate dehydrogenase. Here is an exception that FAD plus molecule is converted into FADH molecule instead of NAD plus molecule. So this is a unique step of Grape cycle where instead of NAD plus molecule, FAD plus molecule is being converted into FADH instead of NADH. So it is a very important step for the production of FADH molecule. Now the fumaric acid is then converted into malic acid, another intermediate of the Krebs cycle, which is also a 4-carbon compound with the help of an enzyme fumarase. Here water molecule is utilized for the production of malic acid. Now malic acid can go into two different pathways. At first, malic acid can go on either to form oxaloacetic acid or it can go to form pyruvate again. So first we will see the oxaloacetic acid pathway. At first, the malic acid gets converted into another 4-carbon compound which is oxaloacetic acid with the help of an enzyme malate dehydrogenase. Here the NAD plus molecule is then converted into NADH just like the earlier steps. And on the other hand, the malic acid can be converted into pyruvate with the help of an enzyme which is known as malic enzyme. And this malic enzyme is very important because it can bypass the pathway or the route of malic acid to produce pyruvate instead of oxaloacetic acid, depending upon the need of the situation. So, in case of malic acid conversion to pyruvate, the malic enzyme is utilized, and here also a NAD plus molecule is converted into NADH molecule, and a decarboxylation process occurs here also because malic acid is a 4-carbon compound and pyruvate is a 3-carbon compound. So, 1 carbon is needed to be eliminated which is eliminated in the form of CO2 molecule. So this is the total Krebs cycle process. So now, at the end of the Krebs cycle, we will calculate the total number of NADH molecules produced. Here, a total of 6 NADH molecules is produced at the end of Krebs cycle. The first one is here for the conversion of isocitric acid to alpha-ketoglutarate acid. The second one is here for the conversion of alpha-ketoglutarate acid to succinyl-CoA, and the third one is the conversion of malic acid to oxaloacetic acid. So 3 molecules are produced at the end of a single cycle, and we know that Krebs cycle occurs 2 times for these 2 molecules of acetyl-CoA. So at the end of 2 cycles of the Krebs cycle, 6 NADH molecules are produced, which ultimately generates 15 ATP molecules at the electron transport system. So then 2 molecules of FADH are produced at the end of the 2 cycles of Krebs cycle, which then produces 3 molecules of ATP, and here 2 molecules of ATP are produced at the end of this Krebs cycle to produce a total of 20 ATP molecules at the end of the total Krebs cycle. And we all know that this Krebs cycle occurs in the matrix of the mitochondria in eukaryotes and in the cytoplasm of prokaryotes. So this is the end of Krebs cycle. So now let's recapitulate the total story of respiration till now. At first, the respiration is a catabolic process by which our body dry weight is reduced to produce energy or ATP. We all know that our body dry weight, which is respiratory fuel, that is glucose, is utilized in the respiratory process or respiration to produce ATP. And the second step, it is written that the glucose acts as the respiratory substrate, just I have told. And in the third, that glucose is converted to 2 molecules of pyruvate by the process of glycolysis. Then the glycolysis occurs in the cytosol of the cell, whereas the mitochondria and the link reaction occurs in the mitochondrial matrix of the cell, but the glycolysis occurs in the cytosol of the cell. Here each molecule of pyruvate is converted to each molecule of acetyl-CoA by the link reaction. Then the link reaction products, which are acetyl-CoA molecules, then enters into the Krebs cycle to produce NADH, FADH and ATP molecules to generate ATP during the electron transport system or electron transport chain or oxidative phosphorylation step. And at the last, the Krebs cycle occurs in the matrix of mitochondria of the cell. So this is the story of respiration till now. Thank you for watching the video. The next video will be on oxidative phosphorylation or electron transport system or electron transport chain and the total ATP calculation of the respiratory process. So if you like the video, please subscribe our channel BiG Science Academy. Click the notification icon for the notifications for the future videos and follow us on Facebook and Telegram. The link is given in the description below. Thank you again.